Alrighty, y'all. So we are going to get started on the final section for, well, final two sections, I guess, for your text. They're both pretty short, so you can get through them pretty quickly. So we'll start with section 3. So this is, chapter 5, section 3, and it's called Other. Scientists compare body structures, development before birth, DNA, and DNA to determine relationships, to determine evolutionary relationships specifically. So we're going to look at similarities in body structure. So first off, we have vertebrates. Now, vertebrates are animals with an internal skeleton that has a backbone. So a person would be a vertebrate, right? A frog has a backbone, would be a vertebrate. A dog, most mammals that we talk about are vertebrates, right? We've got fish, amphibians, reptiles, birds, mammals. These are all vertebrates, right? They all have a skeleton, an internal skeleton with a backbone, okay? They're going to look at homologous structures. These are similar structures that related species have inherited from a common ancestor. So these are two different species with similar structures that are from a common ancestor. So one of the cool things we talk about is the human hand, right? We have 5 digits, and we have a bunch of kind of weird little bones in our hand, in the palm of our hand, that kind of sit next to each other and slide around each other. We talk about our hand, we can compare it to something like a dog's foot, right? The dog also has 5 digits, right? But the dog is walking on their foot instead of using it to grasp things, right? The way the dog's foot hits the ground is very different from the way our hand works or from the way our foot hits the ground, right? Essentially, they're walking on their toes rather than flat. However, the bones, the number of bones and their placement is very, very similar, even though they do very different things. Okay, so similar structure with somewhat related species. We're not super closely related to dogs, but we are both vertebrates, right? And have inherited from a common ancestor. We also look at early development, like embryos. So when we talked about Darwin, and when you saw the Darwin video, you notice that Darwin and scientists around him like to look at embryos. They like to look at baby animals before they're born, right? Or in the eggs, when you saw the scientists looking at the birds and the eggs, they stole an egg, they were trying to look at the beaks of those embryos. So they really look at embryos a lot. We've got similarities in DNA are pretty common. And when we look at similarities in DNA, that's really what we go off of now. We used to look at these homologous structures, we used to look at early development, but that the genetic evidence is so much easier to look at and compare than some of these, because we could be wrong with these, right? This homologous structure that is similar between a cat and a dog, maybe in their leg, or maybe a human and a dog in the leg, that's just looking, right? When we compare the DNA, that's where we get our good evidence. So we're going to talk about combining evidence. Now here's what I'm going to ask you to do. You're going to do a lab online. It's actually a game, which is super fun. And sometimes we've done this before in my class, not you guys, but in previous classes. And sometimes the game gets too interesting and you forget to do the work behind it. And you get really, really far in the game, you get really excited, and then you realize, oh no, I forgot to do the questions. So, game is there, worksheet is at the bottom, and it says first 6 pages only. Don't panic. Each page has about 2 to 5 questions on it, kind of depending on the page. So it's really not as bad as you're freaking out and like, oh my gosh, 6 pages. It's not too bad. So you're only doing the first 6 pages. You can put them on a separate sheet of paper if you'd like. You can print them out and then scan them in if you'd like, or take pictures of them. Whatever is going to work best. So just a heads up for that as well. Please note that each section in the worksheet will actually be labeled. So at the very beginning, the first one wants you to watch the intro lab, or the intro video. So watch that video and then you can answer those questions. The second one wants you to watch the intro video for the first segment, right? And then after that, it starts on the actual game portion. So those first 2 pages are just watching a video and then answering questions, watching a video, answering questions. The other 4 are directly linked into the game. So please just be ready to stop after each round and answer those questions, because if you don't, you're going to have a lot of trouble with this, okay? Again, a lot of people have a tendency to get really excited and play like 4 or 5 rounds, and then they realize, oh no, I have to go back and redo it so that I can answer the questions. So just don't get ahead of yourself. You've got this. Just that like straight up warning for you so you don't get confused or anything, okay? All right, moving forward. So advances in genetics. This is actually one of the more fun sections here. So this is the second section that we're doing today. That first one, again, so much of it can be done through that branching through the branching tree diagrams and all of that. If you're struggling with this, I apologize for getting a little off task. Go ahead and check out page 162 and 163, and it gives you an example of the branching tree diagram. The concept behind branching tree is that we are using it to diagram how closely related different organisms are, okay? And that Noble Lab, that is what that is about. I apologize for not clarifying that previously. Okay, forward. So advances in genetics are 3 methods that people have used to develop organisms with desirable traits. They are selective breeding, cloning, and genetic engineering, okay? Let's talk about selective breeding first. This is the most common process that we use. We use it all the time. The process of selecting a few organisms with desired traits to serve as parents of the next generation. So when we talk about selective breeding, I love to think about fruits and vegetables, right? The original native corn was kind of small, and it was really not like the sweet corn we have, right? It was crunchy, it was mostly animal feed. The corn we have now, when you bite into it, it's very soft, it's very sweet, it's really tasty, right? And that's due to selective breeding. We've bred that corn the way we want it to go. We also have things like dogs, right? Let's take a pug, for example. We got this weird dog with a flat nose and a curly tail. And how did we get there? We selectively bred for dogs with shorter and shorter noses, and as time went on, we got a pug, right? So we're picking the parents, so we're picking the pug parents that look most like what we're going for, and we breed them together and together again and again until we get that pug that we're looking for, right? Inbreeding is our next one. This is bad. It involves crossing 2 individuals that have identical or similar sets of alleles, like siblings or relatives, okay? Last but not least, we have hybridization. This is when breeders cross 2 genetically different individuals. So think crossing a golden retriever and a poodle, right? You get a golden doodle. That is a hybrid, right? They're very genetically different individuals. We're breeding them together and getting kind of cross between those 2, okay? All right, moving forward. Here's the last slide of today. We've got cloning. So an organism that's genetically identical to the organism from which it was produced is called a clone. What I'm going to ask you to do is you're going to watch this video on cloning, and then in the page, in the comments page where this assignment is, you are going to write down one thing about cloning that you previously did not know, okay? If someone else already has written what you wanted to write, please, underneath theirs, comment on it. Say something along the lines of, oh yeah, I agree with you, this is super cool and new to me. Something along those lines, so that you are still engaging, but you're not necessarily writing the same thing again. Last but not least, we have genetic engineering. These are genes from one organism that are transferred into the DNA of another organism. So one of the ones that I like the most is we actually have mice that glow. What they did was they actually took genes from a jellyfish. The gene is called GFP, something fluorescent protein, glowing fluorescent, something like that. And they took this GFP out of the jellyfish and put it into the mouse embryo and let the mouse grow up. And when the mouse grows up, its fur glows. It's actually really cool. So that's the concept behind those guys. Alrighty, ladies and gentlemen, get those two little things done, and that's about it. I believe in you. I know you've got this. Please let me know if you have any questions. Bye, guys.